So today's topic is the nervous system. We already last time talked about some of this basic stuff at the beginning, but we'll do a quick overview. Your general nerve cell is called a neuron. Now when we talk about neurons, we're talking about the cells that are actually conducting the impulses. There are also other cells in your nervous system called glial cells, and these are mostly supportive. These are the conducting cells, the neurons. And what we talked about last time, first of all, were different kinds of neurons. We talked about motor and sensory and interneurons. And how that motor neurons are mostly conducting impulses which way? In which direction? Toward the central nervous system or away from the central nervous system? Away from. So we might say away from the CNS, the central nervous system, to what? To a muscle. And sensory go just the opposite. They're taking information to the CNS from, it's usually some kind of sensory receptor, like a touch receptor in your finger or a cell in your retina. So to the CNS from a receptor. And then interneurons usually connect these two together. Specifically during what kind of setup? When do you want these two things directly connected together? Sensory to motor neuron. Awkward pause. How about during a reflex? And this is what we call a reflex arc. We talked about it a little bit last time. A reflex arc, information goes directly from a sensory neuron. It travels to what part of your body? Look at that big blown up part of your body there. Spinal cord. That sensory neuron is directly linked up to a motor neuron, straight to a muscle. This is what we call a reflex. You don't want your brain involved here because your brain has to do what? It has to process, and that takes some time. Reflexes you want to happen fast, automatic. So this is about speed. If you touch a hot stove, you want to jerk your hand back very fast. You don't want to have to think about it. That's the purpose of a reflex. Sensory neuron to spinal cord. An interneuron between connects the sensory neuron to the motor neuron, and that causes you to jerk back really, really fast. That's a reflex arc. Now, we also last time talked about the parts of most neurons. And those parts, make a little space here. The main part of the nerve cell, the main part of the neuron is this part right here. The cell body, or we've been using the word soma to mean body lately. That's where you find the nucleus and most of the organelles, right there in the middle. But branching out of that, like tree branches, are dendrites. We said that dendra means what? Like a tree. What's the function of dendrites? Yeah, to take in incoming information. Usually that's coming from another neuron, maybe from some kind of receptor. And then usually there's one outgoing extension, and that's called the axon, that carries information out. And it's going to be hooked up on the far end to either the dendrite of another neuron or maybe to a muscle. Those are the parts of a neuron. Now, the other thing that we concentrated on last time was the synapse. And we said over and over again in class that synapse means what? To connect, to join. So a synapse is when the axon of one neuron connects to the dendrite of another. We call this first neuron presynaptic because it's before the synapse. And we call the one on the other side of the synapse postsynaptic. Now remember what happens. As you've got electricity And by the way, that word right there is going to be an important word today, or phrase, action potential. That's the word for the electrical impulse that travels through the neuron, action potential. In physics, what is the word potential? Also another word for. What electrical sounding word? We haven't gotten there for you guys who are in physics right now, but potential is another word for voltage. So that should help you to remember this, hopefully. Voltage, it's electricity. As that electrical signal travels through the neuron, it causes little gates to open up right before the end of the axon. Gates that are specific to calcium ions. So as that impulse gets near the end of the neuron, calcium rushes into this axon. Calcium ions, they rush in. So many of them rush in that they cause these little vesicles to get pushed out. What's inside those little vesicles? What's inside the vesicles near the end of a neuron? Neurotransmitters. So the influx of the calcium ions cause the outgoing outflux of neurotransmitters. And those neurotransmitters get dumped out into the synapse, like you can see happening there. Those neurotransmitters diffuse across the gap, the synapse, and they bind to receptors on the postsynaptic neuron. Now those receptors are of a special kind. If you remember when we were talking about cell signaling, one kind of receptor that we talked about was a ligand-gated what? Anybody remember where I'm going here? Ligand-gated ion channel. That's the name for these little receptors. A ligand-gated ion channel. Now in this example, the ligand is what? The neurotransmitter. And once they bind here to these receptors, these ligand-gated ion channels, what's the message to the channel? What happens after a receptor and a ligand get together? Conformational change. And in this case, what the change causes to happen is those little channels open. Any idea what ion those are channels for? Channels for sodium ions, Na+. And when they open, you're going to get a rush in of sodium into that postsynaptic neuron. Starts to rush in through all these little channels, sodium does. But before we get into the how-to of that, right after these neurotransmitters do their thing, what should happen to them? After they bind to that ligand-gated ion channel, what ought to happen almost immediately to the actual neurotransmitters? They should either be broken down or taken back up into this presynaptic neuron. Because if not, what's going to happen to this postsynaptic neuron all the time? It's going to constantly be firing, right? The message is to fire once and then stop. So the neurotransmitters get released, and then right after they make something happen in this neuron down here, they get broken down or they get taken back up into that neuron. That way this neuron is not constantly firing. All right, let's talk about what's called an action potential. We got a graph here. Most neurons, let's just draw a little piece of a neuron out here. Say this is inside the neuron. Most neurons inside the neuron have proteins in there. They've got some chloride ions. And they also have a bunch of potassium ions in here. Regardless, the number of negatives outweigh the number of positives. So inside a neuron is usually negative. For most neurons, it's usually around negative 70. What's an MV? What do you think MV would stand for? Millivolts. A slightly negative charge. Not very negative. Negative 70 millivolts is not much, but it's a little bit negative. But notice there's lots of potassiums in the neuron. What is there a lot of outside the neuron? Sodium ions. Now, is that the way nature likes things to be? No. What has to happen to keep it that way? What's the only way that nature can build up a gradient? What process builds up gradients? It did it in photosynthesis, it did it in transpiration, active transport, right? Something has to be pumped. And what's the most common example of active transport we talked about in this class? The sodium-potassium pump. That pump inside the neuron pumps potassium into the neuron and sodium out constantly to keep this setup, to keep this gradient. So here's what happens. Go back up here for a minute. Oops. That's not good. So when those little neurotransmitters bind to those ligand-gated ion channels, we said it opens up the channels for what? What do we say those ligand-gated ion channels were channels for? Sodium. Well, if on this neuron there's channels for sodium, right now they're closed, but all of a sudden that neurotransmitter binds and opens those channels, which way is sodium going to go? In or out? If there's lots of sodium out here and not hardly any in here, it's going to go in, right? Well, as sodium rushes in, what starts to happen to the charge inside the neuron? Since sodium is Na+, it starts to go up. And that's what we see happening in this graph, right? We start off at negative 70. As sodium starts to come in, as some stimulus causes those neurotransmitters to bind to the ligand-gated ion channels, sodium goes in. And that causes this to go up. Now, if the stimulus is big enough to get that charge up to negative 55, which is called threshold, it's going to go all the way. If there's not enough stimulus to get it up to negative 55, nothing happens. But if it gets the charge up to negative 55 millivolts, the threshold value, more and more sodium rush in, rush in, rush in, rush in, rush in, and the charge usually peaks out at about positive 40 millivolts. At that point, there are also channels on this nerve for potassium. And most of these channels are said to be voltage gated. Which means when the voltage gets to a certain level, they either open or close. When the voltage gets up to positive 40, what happens to the sodium channels? They close. But what happens to the potassium channels? They open. And if the potassium channels open, which way is potassium going to go? If there's a lot inside and not very much outside, what's potassium going to do? It's going to go out. And as potassium goes out, what happens to the charge? It goes back down. And it goes down to negative 70. It actually goes sometimes a little bit below negative 70. To get back down to that, what we call resting potential. And here's the thing. Notice there's a little undershoot here. When the nerve is not where it needs to be in terms of voltage, it can't work. So sometimes that little period of time there when it can't work, it's called a refractory period. During this period of time, what's the nerve got to do? Can it work? I mean, once we get to this point here, most of the potassium's on the outside and most of the sodium's on the inside, right? Is that the way the nerve needs to be to work? Nope. So what's got to kick in during this period to get things back to normal? Sodium-potassium pump. So the sodium-potassium pump kicks in here, and it's going to pump sodium out and potassium in, get it back to that gradient that it needs to work. That takes some time. So let me go back through that again, because I know that's hard to understand. The most important thing for you to get today is what's an action potential? How does it work? So again, you start off with a neuron with lots of potassium inside and lots of sodium on the outside. There's a gradient, a concentration gradient. But even though there's a lot of potassium on the inside, there's still more negatives on the inside than there are positives. So the neuron starts off with a voltage inside of about -70 millivolts. That's called resting potential. That's what the voltage should be inside the neuron when it's not doing anything, when nothing's happening, about -7. All of a sudden there's a stimulus, something happens. Neurotransmitters cross the gap of the synapse. They bind to those ligand-gated ion channels, and those channels open. And again, we said those are channels for what? What ion? Sodium. As those channels open, sodium rushes in from high to low. And as that sodium comes in, that causes the voltage to start to go up. And like we said, if there's enough stimulus to get it above -55, the threshold voltage, it goes all the way. And it'll go up to like +40. At that point in time, we said, what happens? +40. What happens to the sodium gates? They close. And the potassium gates open because they're both voltage gated channels. When the potassium channels open, there was a lot of potassium inside, not hardly any outside. The potassium rushes out. And that's why this voltage starts to drop back down. So it goes a little bit past where it needs to. And during this refractory period, sodium-potassium pump is getting things back to normal. It's building a gradient back up. It's pumping the sodium back out and the potassium back in to get back the resting potential. And notice this whole thing happens in like a 5 millisecond time period. So we're talking 5,000ths of a second. All that stuff that I just described happens. It took me 5 minutes to explain it, when in actual time it happens in about 5 milliseconds. It's quick, very quick. But this is essentially what generates the electricity in a nerve. This is the electrical generator of the nerve. It's about moving sodiums in and potassiums out. Sodiums in, potassiums out. This is kind of a summary of what I just said. That for an action potential to happen, neurotransmitters from the presynaptic neuron bind to ligand-gated ion channels on the postsynaptic neuron. And again, those channels are channels for sodium. They open. This causes an influx of sodium ions into the postsynaptic neuron. If the potential goes up to -55, the threshold, the actual action potential begins. The voltage-gated ion channels continue to open more and more, which means more and more sodium comes in. Eventually, it'll get up to, on our diagram, it said +40. It can go up to as high as +55. That's when the sodium channels close and the potassium channels open. Potassium comes out, moves from high to low. And that brings the potential back down to -70, maybe a little bit below. And then the sodium-potassium pump kicks in to repolarize the neuron. Bipolar, what do we mean? What's polar mean? We talked about water being polar, we talked about different things being polar. What does it mean that water is polar? Yeah, it's got one side that's negative and one side that's positive. But when we talk about a neuron being polar, we talk about there being one side where there's a lot of what? Sodium, and the other side where there's a lot of what? Potassium. That's what we mean by repolarization. I'm going to take a time out here for a second, and I'm going to show you a little video clip that explains the same thing in a slightly different way. Hold for a minute. All right, we talked a little bit last time about this insulation around the axon. We said that insulation is called the myelin sheath. But notice it sort of looks like those hot dogs that you get in a chain. And there's gaps in the insulation. Notice like right in here there's a gap, and right in here there's a gap, and right in here there's a gap, and right in there there's a gap, and right in there's a gap. Those are called the nodes of Ranvier. And in those nodes, there's a lot of gates for sodium and potassium. So that instead of this action potential actually going through this whole neuron like electricity has to go through a wire, here's what happens. Let's say that the action potential starts here. Well, that causes sodium channels to open over here. And when the voltage gets high enough over here, it causes sodium channels to open up over there. When it gets high enough here, it causes them to open up over here. So what I'm trying to tell you is instead of the action potential having to go through the whole neuron, it's jumping from what? From node to node. And that makes it happen a lot faster. You want your action potential to be really, really fast. So because of this idea of these voltage gated channels, when the voltage gets high enough at this spot, it can cause the channels to open up over here and vice versa. And that causes that kind of conduction is sometimes called saltatory conduction. Essentially, what it means is that the action potential can jump from node to node to node to node, making it way faster than it would be otherwise. So let's go back through this action potential diagram one more time. Make sure we got this. So normally, a neuron, said before, has lots of potassiums on the inside, lots of sodium ions on the outside. And that's what we call polarized. It's got a sodium side and a potassium side. The neuron is polarized. That's normal. Then all of a sudden this stimulus comes along. And that stimulus is going to cause the release of neurotransmitters, which are going to bind to those ligand-gated ion channels. And that's going to open up channels for sodium. Well, there's a concentration difference. And if there's channels open, which way is the sodium always going to go? In, from high to low, right? That's the way nature wants to go. So sodium starts to rush in. And as we said, if it's a big enough stimulus to cause the voltage to go up to negative 55 threshold, it goes all the way. And it's like a chain of dominoes. More and more sodium channels get opened up, which means more and more sodium goes in. Getting that up to about positive 40. When it gets up that high, what do we say happens to sodium channels? They close, and what opens? Potassium channels. Potassium starts to rush out because there's a concentration gradient for it too. And as it rushes out, the voltage drops. Sometimes it drops below the negative 70. We said this period when it's below the negative 70 is called what? It's called the refractory period. Or he called it something a little bit different in that video. Anybody pick up what he called it in the video? He called it hyperpolarization. Regardless, during that period of time, the neuron will work. It can't fire again. But notice that's like 1 millisecond at a time that it can't work. So it's a very short period of time. But that's essentially how electricity is generated in your neuron. This idea of an action potential. The generation and transmission of electricity through your neuron. One more brief thing that I will mention today, and then I will let you go. Let's talk about the basic organization of your nervous system. The central nervous system, the CNS. What two things make up the CNS? What two things make up your central nervous system? The brain and the spinal cord. The rest of your nervous system, the parts of the nervous system that branch off the central nervous system, are called the peripheral nervous system. What's peripheral mean? Like around, outside. So these are the nerves that branch off the CNS, the peripheral nervous system. And that's broken down into several pieces. One division of the peripheral nervous system is the autonomic nervous system. Autonomic sounds an awful lot like what? Sounds a lot like automatic, right? And that's sort of what it is. Notice what it says here. The autonomic nervous system is the part of the peripheral nervous system that acts as a control system, maintaining homeostasis. This part of your nervous system is unconscious. You're not thinking about this to make it work. This is an unconscious, innate part of your nervous system. It's keeping things under control. I mean, you don't have to think about, hey, my blood pressure's too high, I need to lower it back down, right? Hey, my heart's beating a little too fast, let's slow it down. You don't think about that. That's unconsciously controlled. The ANS, an autonomic nervous system, controls everything from heart rate to digestion to your rate of breathing, your respiration rate, how much saliva you have in your mouth, how much you sweat, the diameter of your pupils, when you urinate, when you're aroused. It controls all those things. Unconscious things in your brain. Now, we're going in divisions here. So the autonomic nervous system is a branch of the peripheral nervous system. Well, that itself branches. And there's two branches of the autonomic nervous system. They're called sympathetic and parasympathetic. We'll kind of have a flow chart of this next time. I want to look at this real quick. So here's your autonomic nervous system. And you see that most of these parts have two different sets of nerves going to them. Over here we've got let me get myself straight here. Over here we've got the sympathetic on this side and the parasympathetic over here. Let's look at the effects of the sympathetic nervous system on your heart. Notice when the sympathetic neurons stimulate the heart, it speeds up. When the sympathetic neurons stimulate the airways, they dilate. When they stimulate the salivary glands, they inhibit the formation of saliva. When they stimulate the pupils, they dilate. When they stimulate the tear ducts or the tear glands, tears are inhibited. All those things are associated with what? What have we talked about? All those things happening at the same time. What response? Fight or flight response, right? But notice on the other hand, the parasympathetic nerves, when they stimulate the heart, they cause it to slow. When they stimulate the respiratory structures, they cause the bronchioles to constrict, the airways to get smaller. When they stimulate the salivary glands, they stimulate them to make more saliva. They cause the pupils to constrict. They cause the formation of tears. What I'm getting at is that most of the time, both of these systems are working together, keeping you where? Homeostasis. Now, there's certain cases where you want this to be dominant. Cases like when you're scared, fight or flight kinds of times. There are other cases where you want this to be dominant. Maybe you're just relaxed and taking it easy. It'd be nice for your heart rate to slow a little bit, for you to not have to breathe as fast, start making more saliva. You're probably preparing to do what if you're making more saliva? Eat, digest. So most of the time, both of these two parts of your nervous system are working together in tandem. In some cases, though, like I said, this side might be more dominant. In other situations, this side might be more dominant. The last slide we'll look at today is this one. Again, we've got sympathetic over here on the left, parasympathetic over here on the right. This is looking at the effects on digestion. For example, notice that if the sympathetic neurons stimulate the intestines, they cause the activity of the intestines to slow down. They inhibit activity of the kidneys, the stomach, and the pancreas. But on the other hand, if we look over here at the parasympathetic, they increase intestinal activity, increase activity of the stomach and the pancreas. So sometimes the parasympathetic nervous system is thought of as sort of like the take it easy nervous system. It's going to be dominant when you're relaxed, you're calm, you're trying to digest your food. On the other hand, the sympathetic nervous system is sort of like crisis nervous system. Something's bad or wrong. Digestion can wait. You better be preparing for fight or flight if the sympathetic nervous system is dominant. Notice that the sympathetic causes your liver to release glucose. The parasympathetic, on the other hand, causes the gallbladder to release bile. Anybody know what bile does? Nope. It helps you digest your food better, specifically fats. So this side increases digestive activity. This side decreases digestive activity. This side prepares you for fight or flight. This side prepares you to kick back and relax. Most of the time, though, they're working together. They're keeping you somewhere in the middle. Homeostasis. Well, that's a good place to stop. Go home and hope it doesn't